welcome back to Intro to Biology for Majors, or BIOL 1224. This PowerPoint will cover Chapter 19 of your Openstax Biology 2E book. This is from Module 4 of our course, and this chapter is over populations. So as always, we're going to start with our objectives. The first is to recognize measures of population evolution. The second is to identify 4 major contributors to population genetics. And the third is to recognize the significance of sexual selection. So let's start off talking about time. So eons and eras are just 2 words used by scientists to describe the geological time scale. So scientists will use the geological time scale to divide the history of the Earth into both eons and eras. And these periods are defined by major geological or biological events, like mass extinctions, and that's an example like what killed off the dinosaurs. So researchers analyze fossils, anatomy, and molecular sequences to learn how different species are related to one another. So the whole point of this PowerPoint is to really talk about populations. And in this slide, I want to talk about population evolution. So population evolution describes the evolution of populations and species from small-scale changes among individuals to large-scale changes over paleontological periods of time in an entire population. So to understand how organisms evolve, scientists can track the population's allele frequencies throughout time. Scientists track with this statistic over time to measure overall change in these populations. So let's talk a little bit more about the geological timescale. So a couple of previous slides ago, we learned that scientists divide the history of the Earth into both eons and eras. And to do that, they define major events like mass extinctions or the Ice Age, for example. So researchers will analyze fossils, anatomy, and molecular sequences to learn how species are related within these time periods. And a fossil is actually defined as any evidence of an organism from at least 10,000 years ago. So if anything that we find is younger than 10,000 years, it's actually not considered a fossil. So in this slide, we're talking a lot more about fossils. So fossils can form in many ways. Plants, impressions, resin, footprints, and organic matter can all form fossils. They preserve evidence of many types of organisms from our past. The fossil record that we currently have is actually incomplete, and the fossil record meaning all of the fossils that we've found up to this point of these different eons and eras. So organisms like jellyfish that have soft bodies will fail to fossilize. So we'll never actually have a complete fossil record based on that fact. And there's an image here at the bottom of your slide of an ammonite. So this ammonite, it's actually a marine fossil, and it indicates that at one point Oklahoma was covered in water, much like an ocean, because this ammonite was actually found by paleontologists in Oklahoma in Lake Texoma. So this other image on the top kind of shows you the process of fossilization, and in this case it looks like they've got a dinosaur up here. So for example, you could always have fossilized footprints, impressions. They're left in the mud, the dinosaur itself collapses and dies, the flesh will rot away, the bones remain, and then over time water level will actually rise, sediment buries the bones and footprints themselves, and a thick sequence of sediments or dirt and rock accumulates over the bones. Gradually, the bones will become fossilized, so almost like a stone. This bed contains the dinosaur bones, and then erosion will eventually expose the layer of strata containing the bones, and paleontologists are able to find these bones this way. There are lots of dig sites, even in the state of Oklahoma, that paleontologists go to find dinosaur bones to this day. So this slide really helps us discuss determining relationships in organisms throughout time. So relative dating is a process that scientists use. It's a term, and it basically assumes that the lower the rock layers, the older the fossil that they find, and the newer rock layers that we explore, the younger the fossils, which makes a lot of sense. The deeper that you are in the ground, the older those fossils are going to be. So now I kind of want to talk about homologous structures, which is really going to tell us, how things are related over time. And this top image, the top left image, will kind of show you the top right will show you what I'm talking about. So in this image, you see a human arm, a cat's leg, a whale's fin, and a bat's wing. And in this image, you can see through them, you actually see the bones located within these structures. And if you look, we have a humerus, a radius ulna, carpals, metacarpals, and phalanges, which are the bones that make up our arm. Actually, cats, whales, and bats all share very similar structure within their given arm, whether it be a whale's fin or a bat's wing. We all have very similar structures that perform similar functions. And please remember that all of these are mammals, right? So we're a mammal, a cat is a mammal, a whale and a bat are mammals. Even though they swim and fly, they're still considered mammals. And these homologous structures, or the structures that all look the same and perform a similar function, actually reflect a common ancestry in our mammalian roots. So this is a result of selective pressure. We did discuss selective pressure when we talked about the finches and Darwin. So basically, over time, mammals have evolved to have different functions for survival purposes. So different than analogous structures, which may be similar but have no common ancestor. So an analogous structure would be, for example, wings of birds and bees. So birds are, obviously, they have wings that help them to fly. Bees also have wings, but bees are insects. Birds are not. So they have a very similar function, but they have no common ancestor. And I also like to talk about vestigial structures, which is very interesting for some students because I feel like a lot of people don't realize. We know that whales are mammals, but evolutionary time has shown us that actually whales at one point did have legs and pelvic bones, and they were terrestrial. They did walk on land. So vestigial structures are structures like this vestigial pelvic bone that is still found in all whales today, and they have lost their function. So these hind limbs, the whales no longer need because they survive solely in water. But at one point in evolutionary time, within the eons and eras that we discussed earlier, whales were terrestrial. They did walk on land, and then they also could swim. And over time, they lost these vestigial hind limbs because they didn't need them anymore. And also, I'd like to say that DNA is used as a molecular clock. So basically, we can look at our current DNA of either ourselves or other organisms and kind of look back at the fossil record and fossils that we find and determine the evolution that's taken place in a clock-like manner. So we can look at it over time itself. So now I'd like to talk about some of the factors that change allele frequencies over time. So 4 of those factors that we're going to discuss within the PowerPoint are mutilation, founder effect, bottleneck, and migration. Okay, so let's start off with mutation. So DNA changes drive diversity in populations. So the changes within organisms' DNA drives the overall diversity in populations. And we've kind of talked about this in previous slideshows as well. So the appearance of new mutations is the most common way to introduce novel or new genotypic and phenotypic variants. And remember that genotype is within your body, it's your DNA, and phenotype is what you look like, what's appearing on the outside. So basically they're saying that the appearance of new mutations within individuals that then kind of incorporate into a population is the most common way to introduce new genotypic and phenotypic variants or diversity. So some mutations are unfavorable or harmful. They're quickly eliminated from the population by natural selection, which we've also previously talked about. So basically natural selection and evolution, they don't get to choose anything. Everything is random, right? So some mutations will happen within an individual that are unfavorable or harmful to that individual. And so they won't continue to breed, which would then kill off that unfavorable or harmful mutation. Others are beneficial, so other mutations are helpful, and they do spread throughout the population over time. So whether or not a mutation is beneficial or harmful is determined by whether it helps the organism survive to reproduce. That's the whole point, is surviving to reproduce. So some can have a dramatic effect on a gene and the resulting phenotype. So the resulting outside of that organism will be very different. So in this image, I just show you a few flowers that have evolved different colorations. So you've got yellow up here, you've got orange down here. And they're actually the same species, but evolution has allowed them to change their color. So that gene has changed within them, resulting in a different look, a different phenotype. So now let's talk about a few examples of mutations. So genotypes impact phenotypes, which makes sense if you break it down. Genotypes, or the changes at the genetic level within our genes, will impact phenotypes, or what we look like on the outside. So, for example, the FOXC2 gene actually gives people extra eyelashes. There's a gene called BHLHE41 called for short sleepers. It basically says you don't need as much sleep to function. And there is also a gene called LRP5 that increases bone density, and these are just in humans. So the example that I give with this image is actually Elizabeth Taylor. She had a genetic mutation of the FOXC2 gene that we talked about, which gave her actually an extra row of eyelashes on her eyes. So now I would like to talk about the founder effect. So this may be something that you haven't heard of before. It's actually pretty interesting. So when only a few individuals establish a new population, the allele frequency might change. So this process illustrates the founder effect. And if you think back to Darwin and the finches that we talked about, whenever those finches migrated, a few of those finches of the entire population migrated to the island of the Galapagos and then were cut off from the rest of their population, they formed a founder effect. So there were only a few of the individuals established there for that new population. So the frequencies of alleles within the entire population weren't present. It was just those few individuals that were on the island. So this is what a founder effect is. And that's what I have here, is that the Galapagos finches displayed the founder effect. So this image here kind of illustrates what I'm talking about. So here you have an origin population. So in our founder effect example of the finches, this would be all the finches on the mainland. So the ancestral population, and this is red, it's half and half, and then the green here is just two different, basically, types of the same population. So this is the entirety of those finches, and then we have just a few of those make it over to the island of the Galapagos, and over time, the red frequencies of alleles that over here were evenly spread out have now sort of taken over. So that founder population has made all of the red alleles in these 5 generations just multiply over time, and the green allele sort of died out. So in this example, those red alleles must have been beneficial for the new environment that these finches may have joined. So basically, this is a visual example of a founder effect, and if you want an example that we've previously talked about, it's those Galapagos finches on the island of the Galapagos. So now let's talk about a bottleneck. So a population bottleneck will occur if a disaster drastically reduces the size of a population. And one example that they provide is when animals are hunted to near extinction or to extinction completely. So in the image here, you see an actual bottle they use for an example. So this would be the reason we call it a bottleneck. And in the bottle, they have the original population of the organism we're talking about. Here is the bottleneck. So it's the actual tip of the bottle. It's an event that knocks down the population drastically. And then the surviving population has very few of the original alleles that were within the original population. And so the evolution that takes place from here on out is going to be based on the surviving population, these individuals themselves. So a bottleneck results in a large portion of the genome, which was in this original population, suddenly being wiped out because of a disaster. The genetic structure of the survivors becomes the genetic structure of the entire population. So these few survivors that are left, their genetic structure moves on to all of the subsequent population, all of the new generations. And this may be very different from the original population because we don't share all of the original alleles anymore. And the type of disasters we're talking about are, for example, hurricanes, tidal waves, things that basically knock out a large portion of a native population within a certain area. So now let's talk about migration. So we're all aware that there are several species of animal and other organisms within our environments that migrate. So migration moves alleles between different populations. So this might affect the allele frequencies in both of those populations. So populations will actually decline when the death rates exceed the reproductive rates, which makes a lot of sense. So I like to use the example of birds. And over here, we're kind of talking about birds in this image. These are all different types of birds. We have a hawk here, we have a falcon, a tern. So these are their main migration patterns. This is the way that they migrate. So within this process, the alleles or the genes that these organisms and individuals carry from maybe their initial population are transferred between different populations. So when they arrive in their end goal area, they breed most likely, and those new alleles from the different population are now introduced to the new population. And then obviously it makes sense when populations decline because death rates are extremely high, they will exceed the reproductive rates. So there are more individuals dying off than are being born. And so that's a good example of populations declining, which can happen during migratory processes. But migration will once again move alleles between population. So natural selection results either from competition for access or from one sex choosing attractive mates over another. This happens a lot in bird populations. So basically generations of what we call choosy females have selected for males with, in birds, for example, nest building traits or elaborate ornamentation, basically what they look like, whether they're very beautiful on the outside. And genetic fitness is measured as the offspring produced by an individual that has survived to reproduce. So basically the success of these ornamental or well-building birds is then determined by their offspring and how often they are able to reproduce. So let's talk a little bit more about sexual selection, which is what we spoke about a little bit in the last slide about migration with birds. So sexual selection results from variation in the ability to obtain mates. So in birds, for example, like we previously talked about, ornamentation, what they look like on the outside, as well as their ability to build nests, or in a penguin's case, their ability to be a good parent. So actually, females are seeking out males that they feel will be the best father figure, because the fathers actually maintain the egg once it's laid by the female. So yeah, sexual selection also has a very drastic ability to change the allele frequencies, moving on to subsequent generations. So now let's talk a little bit more about reproductive success, which just builds on the slides about sexual selection. So once again, in birds, for example, complex nests and flashy plumage will lead to sexual or reproductive success. And for example, in mountain goats, butting heads will lead to reproductive success because they look like the toughest male when they win. And all of these things do appear to waste energy, but they lead to reproductive success. So natural selection will allow for traits that apparently reduce survival. So it's not always the positive traits moving on. And sexual selection results either from competition for access, which in the butting heads example would be what we're talking about, or from one sex choosing attractive mates of another sex. So for birds, that would be the flashy plumage or being a good parent. Basically, they choose the sex that they're attracted to. And generations of females, often that are choosy, have selected for males with better nest building traits, better ornamentation, the toughest male that there is. And so they've sort of been able to determine the subsequent generations because those alleles are passed down through those generations by reproductive success of these males. So now let's talk a little bit about genetic fitness. So the total set of gene copies for all genes in a population is referred to as the gene pool. And the gene pool gets that name from the idea that we are essentially taking all the gene copies for all genes in the individuals of a population and dumping them into one large common pool. But that doesn't necessarily determine genetic fitness. So genetic fitness is actually measured as offspring of two individuals produced by an individual that survive and to reproduce. So basically, as these babies or as these new offspring are able to reproduce and move these alleles down the evolutionary line, genetic fitness of the original parents is determined. So in genetic fitness, reproductive success plays a very big role in the slide that we just talked about, in the genotype or the genes and the phenotype, the look of the offspring. So for example, here we have genotype being homozygous dominant, where we have a bird here with a large beak and high fitness. And then here we have a heterozygous, which would be a medium-sized beak with medium fitness. And then here we have a bird that is homozygous recessive with a small beak and another relatively high fitness. So it may not be the obvious choice that the small beak is higher in fitness than the medium-sized beak, but it does happen. So in summary, Natural selection cannot produce the perfect organism. Natural selection can only select on existing variation in the population because it does not create any variation itself from scratch. Basically, these mutations are determined by natural selection to either be beneficial or not, and it is limited by a population's existing genetic variants and whether new alleles arise through mutation and gene flow. So like I said, when these mutations and gene flows change, new traits, whether they're genotypic or phenotypic, arise, and natural selection sort of determines whether or not they are beneficial or harmful. And often both will move forward, but most often beneficial things will move forward because individuals will select for mates with beneficial traits. So here in the image on the left, I have an image of those finches that we talked about through Darwin and the Galapagos Islands. So we have the ancestral finch. Over natural selection, we have multiple different types, and there were many more than 3 on the island, but in this case, we have examples of an insect-eating finch, a woodpecker-type finch, and then a seed-eating finch, because this finch had to evolve to survive. So multiple generations evolved that were able to eat different things on that island, or else if they all ate the same thing, their food source would run out. So this is just an example of natural selection kind of helping to determine the beneficial traits for these finches. So now let's talk about our key takeaways. So number 1, scientists use genotype or DNA and phenotype, which in this example is fossils, anatomy, embryo structure, how something looks, to establish relationships. Number 2 is mutations, founder effect, bottleneck, and migration all contribute to population genetics. And number 3 is that sexual selection impacts reproductive success and genetic fitness. So this is actually the end of the Chapter 19 PowerPoint, and it should be the end of Module 4 for this course.